Hello, this is Barry Moore from Trinity College Dublin, welcoming you to this bite-sized bio web seminar, which today is sponsored by Novus. Novus offers directly conjugated antibodies to measure everything from uncommon targets and less common species to all the most widely studied CD markers in at least 14 conjugate options each. They do this to make optimized multicolor staining easy. Today's presentation is entitled "There's No Business Like Flow Business, Basics of Flow Cytometry," and is being presented by Dr. Rachael Walker, Head of Flow Cytometry at the Babram Institute in Cambridge in UK. Rachael has over 12 years of experience in flow cytometry and has run flow court facilities for over 9 years. As always, we'll have a question and answer session after the presentation, so please type any questions that you'll have into the questions box, which appears on the right-hand side of your screen, and I'll put them to Rachael at the end. So now, over to you, Rachael, for the presentation.
Thank you, Barry. Welcome, everybody. Today in the webinar, I would like to go through how flow cytometers work in order for you to get the most out of your experiment, so that you realize it's not just a box that's in your lab. I hope to show you how the increase in popularity of the technique and how it's evolved. So let's start at the beginning. Let's start with the definition of flow cytometry. It comes from the Greek. We're looking at flow, so that's cells that are actually flowing through a machine. And the cell comes from the cyto, and the metry is the measurement, so it's a measurement of flowing cells. If we look at this definition from the current protocols in cytometry, flow cytometry is a technology in which a variety of measurements are made on cells, cell organelles, and other objects suspended in a liquid and flowing at rates of several thousand per second through a flow chamber. So it's important to know that we are actually looking at cells in suspension. It's not just cells, we can look at other things such as beads, other particles, and we're looking at several thousand, so we can get quantitative data from them. So as I was just saying, we're looking at thousands of events in a short period of time. We from that can get lots of statistical information. We can run 15,000 events per second, so we can easily get millions, billions of cells through our machines and look for rare populations. We can use them for phenotyping, so we can label our cells with antibodies with a fluorochrome attached to them, and then we can look for subpopulations of cells. We can also look at the DNA analysis, so we can look at the cell cycle of the cells by using dyes such as beryllium iodide. We can also look at functional studies, so we can look at the calcium flux of cells. With flow cytometry, we've got lots of flexibility in what we can do, and the machines are capable of doing more than one application, so we can actually look at lots of different parameters simultaneously. We also have the ability to reanalyze our data so that we can get new information. So this can be very useful if you're doing a multicolor experiment and you find a subpopulation that you didn't realize was there. You can then go on to look at that population in your studies you've already done. Often you will hear flow cytometry also known as FACS, but many people don't understand what FACS is and the difference between that and flow cytometry. FACS actually stands for Fluorescence Activated Cell Sorter, and it is a trade name. It is properly known as flow cytometry. It's a trade name by Becton Dickinson, and it's like saying that you're using a Hoover or a Biro or Google. It is actually more technically correct to use the term flow cytometry. Flow cytometers can be both analyzers and sorters. For the analyzers, we look at the cells, we look at the fluorescence that's emitted, and then the cells go to waste. Whereas in a sorter, we use the same principles as we do in an analyzer. We then can separate out the cells to do further studies with them. As you can see from this chart, the popularity of flow cytometry has increased in recent years. We start in the mid-1970s. That's when the first commercial cytometers were available. Since then, we've had an increase behind the technology of the machines, behind the lasers and the fluorochromes, which has meant it's been possible to get more data, different applications, and therefore more publications. Here are some of the commercially available machines. So there were machines that you found in the clinical lab, there were machines that are found in the research lab, and there were also machines that have varying degrees of complexity. There were some machines that are made, such as the Curie or the FACS Caliber, which are simple 2-laser 4-color machines. But then we can get the Fortessa from BD, which has 18 colors. There are also some other machines that have slight differences to them, such as the Amnis image stream that takes an image of every cell as it is going through the cytometer. Here is a picture of a sorter, which is actually from Babram Institute. This was taken in 1987, and it shows that our machines nowadays are a lot different. They're a lot smaller, and the technology has improved. So actually, what that machine could do then, we can actually now do in a very small machine. In order to understand how we get the machines in our lab today, we can look back at some of the history of them. Wallace Coulter in 1949 invented the Coulter Counter, which is actually still found in many hematology labs today. It looks at changes in electrical resistance when cells pass through an orifice in order to count and size the cells as they pass through. Mack Fulwyler in Los Alamos was using one of these machines and was looking at red blood cells, and he could see two different populations, and he decided he'd like to see if there's a difference between them. So he invented a cell sorter. This was done in 1965. He used the principles of inkjet printing from a paper by Richard Sweet. So what it does is charges a droplet of solution containing a cell into a pot. Len Herzenberg, who was an immunologist, took the idea of this technology and added lasers to the machines in order to excite the fluorochromes that were attached to his antibodies. This then led to the first commercially available machine, the fax machine, in 1972. For the rest of the talk, I want to talk about how we go from cells being in a tube to going through a machine to give us results on the screen, but then we'll hopefully give you the paper you've always wanted in Nature, and then a standing ovation in meetings. So let's talk through how the machine works. Quite simply, we start with a cell population. We want to have single cells, so it's very important that we have the cells in a single cell suspension. We pass them through Fluidics to the machine. They get interrogated in the flow chamber by a light source. We then can detect and process any signals that then go through the machine for analysis and are displayed on our computer. So let's go through the different components of the cytometer. We're going to start with the fluidics. This is the separation and the alignment of the particles through the machine. If we were to sort, it would include the sorting part and how we can actually adjust the speed that the cells go through the machine. We've got the optics. This is the light sources that we use to excite the fluorochromes, how we detect the photons that are emitted, and how we separate the light through the machine using a series of filters. We want to look at the electronics. That's how we collect and analyze the optical signals, how we can display the data, and then we look at how we can analyze it, how we can look at it on the screen. Most flow cytometers contain sheath fluid, and this sheath fluid is usually PBS, and it's used to deliver the cells from a tube to the flow cell of the machine. And what it's doing is it's actually aligning the cells so they can go through the machine, so they go through the center of the laser beam where the laser beam is the most intense. The cells, the sheath fluid, don't actually mix, so actually some cytometers can be run using water. Most of the machines that are on the market are actually differential pressure machines, for example the BD fax machines, and this is where the sample and the sheath fluids are pressurized. We can control the flow rate by changing the sheath pressure, and we can control the difference between the sheath and the sample pressures. However, it's not possible to do absolute counts. This means that if you're wanting to know exactly how many cells are in your tube, it is not possible to do this accurately on a differential pressure machine, and you should use some sort of enumeration beads. So when your cells are put onto the machine, they are pressurized and they go through the sample injection port into the flow cell. And within the flow cell, their cells are hydrodynamically focused by a sheath fluid so that they come out one by one. So the hydrodynamic focusing actually pulls the cells out like a string of pearls to ensure each cell goes to the laser beam one by one. Here's a little animation of how this works. So if we are running our cells on low speed, what we actually have is we have a tight core of sample in the middle, and the cells are going through the middle of the laser beam. However, if we turn the machine onto high, we're actually more likely to have two events going through the laser beam at the same time, and this can cause errors and problems with your data. If you're ever doing cell cycle, it's very important to have them on low and not to go to high. So one of the hints I have for you is to have more concentrated samples and run them on low. With the fluidics of your machine, if you don't have a clean fluidics, it can cause quite a lot of problems with your experiment. So this animation shows that cells can get stuck to clumps, and it can cause problems where the cells aren't going through the middle of the laser beam. Here's a few hints on what could be wrong if you've got some problems on your machine. If you don't see any events on your screen, it could be that you've got air in your system, your fluidics cart's not switched on, you could have a cracked tube, so therefore the sample's not being pressurized. You also want to make sure that you have the machine set on low, medium or high. Have a check to see if there's any difference if you go to the different settings. It could be that you've had a software crash, or it could be a blockage. And a good way of seeing if you've got a blockage is to run some hot water. So just get some water out the tap and run that. And that sometimes can help to move a blockage. If your tube is filling up, it could be that you've got back pressure in your system. If you see that your cells are dancing around on the screen, it could be that you've got a partial blockage, so you've got some turbulence happening and your cells aren't going through the middle of the laser beam. You also could have your cells too concentrated, so it's very important to make sure that you have your cells at the optimal concentration. So now we've talked about the cells going through the laser beam, getting to the laser beam, let's have a chat about the optics. So the optical path is from the interrogation point in the flow cell to the detectors, and we have filters and dichroic mirrors in there to separate out wavelengths and to drive them through the different pathways. So most cytometers will have a laser as a light source. Not all of them do. Some of them have LEDs, some of them have mercury lamps, but a majority have lasers. A majority have more and more lasers as the technology behind lasers has improved, and we're now getting smaller, more stable lasers. So we use lasers because the light is coherent, it's tight, defined, with little divergence. We also can get single wavelength of high intensity light from them. With your cytometer, it's important to know how your lasers are in the machine. Are they collinear, where two lasers are sharing the same path? Because this could limit what fluorochromes you could use together. Or are they spatially separated, where there's a space between each laser? If we look at the laser beams, what we're in machines is we tend to use near enough the full spectrum. So most cytometers have a 488 or blue laser. That's usually the primary laser. Most cytometers also have a 635 or red laser, and it's commonplace to have a violet laser there as well. In the last few years, there have been more and more machines available with a 561 laser, which is used for red fluorescent proteins, and also machines can have a UV laser. So looking at the spectrum, it's important to note that the shorter wavelengths have a higher energy and the longer wavelengths have a lower energy. So when we've got fluorochrome in our cells, we're going to excite the fluorochrome and it will fluoresce. So what we want to do is excite that fluorochrome as close to the excitation maximum as possible so that we can get the most efficient excitation. The excitation will actually also occur at wavelengths near to the excitation maximum, it just won't be as intense. The same when we actually want to collect that fluorescence, we also want to collect the emission as close to the maximum as possible so that we can get the best data from our cells. So we always want to try and match our fluorochromes to the best laser and the collection filter to the emission maximum. However, it is possible to collect fluorochromes wavelengths near to the emission maximum. So the top graph there shows that illumination at higher or lower wavelengths only affects the intensity of the emitted light. Therefore, if you haven't got a laser that's going to be exactly at the excitation maximum, it's fine, you can still get some results from them. The emission wavelengths that are given out in fluorescence is always lower than the excitation wavelength. So therefore, there's a Stokes shift. Each fluorochrome has a distinct Stokes shift. For example, FITSI in the top graph gets excited by a 488 laser and emits around about 530 nanometers, and this is the Stokes shift. As each fluorochrome has a different Stokes shift, it's possible to use multiple fluorochromes from the same laser, and therefore we can get the most out of our cytometer. So what parameters can we detect? We can detect scatter. So this is when the cell goes through the laser beam and it scatters the light of the laser beam. Straight ahead we collect forward scatter. This is sometimes incorrectly told to people as being the measurement of size, but it's not a true measurement of size, as we have to take a lot of other factors into consideration, such as the solution that the cells have been in, the cells themselves, whether or not they've got a more blebby membrane. If you were to put a 10 micron bead and a 10 micron cell through the cytometer, you'll get vastly different forward scatter. So it's important not to think that your cells have got bigger or smaller just by looking at the forward scatter. If we look at side scatter is the internal morphology, so that light that's scattered from inside the cell, and we collect that at 90 degrees. And what we can do is we can look at the forward and side scatter together to be able to distinguish cell populations. So we can look at lymphocytes, monocytes and granulocytes based on their differences in forward and side scatter. We also collect the fluorescence at 90 degrees along with the side scatter. And what we're actually detecting is we're detecting photons of light that have been emitted during fluorescence. So within the machine we have a series of optical filters that we use to steer those photons of light to the correct detector. So here's some examples of the optical filters. So if we've got a long pass filter, this is a long pass 500, will allow light longer than 500 nanometers to go through it. This is a short pass filter. This will allow light shorter than 500 nanometers to go through it. And then this is a band pass filter. For this, the number is the middle that it will allow through. So it will allow light of 500 nanometers. And then the number to the right, we divide in two. So it will allow light from 575 to 525 to go through the filter. We also use dichroic filters at 45 degrees. And this allows light above the cutoff to be transmitted, and light that would have been blocked is reflected at 90 degrees. So if we look at how these fit into a cytometer, we can see the cells are passing through the laser beam. We're detecting the forward scatter, and then at 90 degrees we're collecting side scatter and fluorescence. At the first long pass filter, the 500, it's allowing light longer than 500 nanometers to go through, and light shorter, the blue light is going through to the detector. And in front of the detector is a 48810 band pass filter, which is allowing the light to go through to collect the side scatter. And the next one is showing a 560 long pass, which is allowing green light to go through towards the 53030 filter. So this would be where we would collect something like FIPSI or GFP. The next one is steered towards the detector. That's what we'd use, for example, for PE. And then for the red, we're using a 670 long pass to direct the red light, and that's where we would collect things like PE size 7. Different cytometers have different pathways, so it's important to know the pathway of the machine that you're using. So BD machines tend to use an octagon where the longest wavelength of light is collected first, because this wavelength of light actually has the shortest energy. Machines such as the Beckman-Coulter Gallios collect the shortest wavelength of light first. So once we have the photon of light being steered towards the detector, we now need to talk about the detection of those photons. And we use a variety of detectors within a cytometer. We can use a photodiode. That's used for forward scatter. And that's when we're looking at a very bright signal. We use PMTs, or Photomultiplier Tubes, to look at fluorescence and side scatter because it's a much more sensitive detector. And some machines have other detectors such as CCD chips or avalanche photodiodes. So how the PMT works is it's actually going to convert your photon of light to an electrical signal. So the photons hit the cathode, which emits electrons, and this is accelerated via a series of additional electrons. It's amplified, and then the signal is collected at the anode. So when you're changing the voltage on your cytometer, you're actually changing the sensitivity of your PMT. It's nothing to do with the laser power. It's all to do with the sensitivity of your PMT. So we've talked about cells going through the machine in the fluidics and how they've gone through optical filters and how they've been detected in a PMT. Let's now talk about the electronics and how we can see the data. So our filtered photons are collected and they're multiplied by our PMT. That current is then generated, goes into a preamplifier where it's amplified further and converts to a voltage pulse. This pulse goes to an ADC to be digitalized, and those values are found on a list mode file, which is a .fcs file. So just to summarize it, we have a cell, we excite it with a laser beam, it fluoresces, it emits photons, they go through a filter to a PMT. Within that PMT, the photon is converted into an electrical signal, and we see that as a pulse on the screen. So this is how we detect an event. As a cell goes through the laser beam, we get the beginnings of a pulse going through the machine. As the cell is right in the middle of the laser beam, we get to the top of the pulse, and as it leaves the laser beam, we get the completion of the pulse. And from this, we've got the height and the width of the pulse, and this happens in every channel that you're looking in your cytometer. In machines such as LSR2 or a Fortessa, what the machine's actually doing is it's taking segments of that pulse and looking for the area. So that if you are going to be using those machines, the area is the most accurate channel to be using. But how do we determine that an event is real? So we trigger on a particular channel, and we actually want to be able to set a threshold, as normally we do that on forward scatter. When we are looking at our sample, we actually could have electronic noise that's in there. And what we want to do is be able to say that the events we're looking at are real. So here's an example of some pulses. So you can see we've got some very small pulses. And we need to have a background threshold so that we are not seeing electronic noise. But where this threshold is below the threshold, the electronics are blind to that. Therefore, if you put it in the wrong place, you're never going to get that data back. So you can see here we've got a whole blood sample. As we increase the threshold, we can see that we've cut off some of the cells. If we take it higher, we're just looking now at 2 cell populations. But if we take it even higher, we've cut off part of those cells. So my advice to everybody is to have the minimum threshold and only increase it if you need to. What you don't want to find out is that you've actually taken out a population you were actually interested in, because you can't get that data back again. So this is a list mode file. This is what your data looks like when it's been through the cytometer. So it has a list of keywords. So it can have like your sample ID. It tells us the parameters we've looked at, the channels we've looked at, and it also has information about every single cell that's gone through the machine. So the flow cytometry files are saved as a .fcs file. What this list mode file actually does is it takes information about each cell one at a time and puts in there the information about where it is on the scale. So this is an example here where we would have cells and it will tell us where it is on the scale, the forward scatter, the side scatter, and then 2 different sets of emission. And what happens is we can actually plot the position of those cells on to a dot plot. And you can see here we start to build up a dot plot until we've got dots on the screen, which is showing us exactly where the fluorescence of those cells were in those channels. And this is just a reminder at the bottom of what a histogram does. So on the left we have cells that are not positive for a FITSI marker, and to the right we have the cells that are actually positive for FITSI. And you can see towards the right the intensity of the fluorescence increases. What does our data look like? We can actually see each cell on the screen as a dot. We can look at them as a histogram or dot plot. So on the left-hand side you can see that we can look at one parameter at a time on a histogram, but we actually get a lot more information if we were to put that together to see a dot plot, where we can actually see how populations are related to each other. We can see data using either a linear or a log scale, and this depends very much on the application you're going to be using, which scale you will use. We usually look at forward and side scatter on a linear scale because we're looking for a very subtle difference in the amount of scatter. We tend to look at fluorescence in log because there's going to be a big difference between a cell that doesn't have a fluorochrome on there and one that does. So we tend to use log. If you're going to be doing cell cycle, you do that in linear, because it's a very subtle difference between cell in G0, G1 and in G2F. So you might be asking yourself, why do you need to know all of this? You know, you're not going to be designing a flow cytometer anytime soon, but actually it's really important to know how to use your flow cytometer. It will help you to design your experiments better. It will help you to realize what you can actually do with your cytometer. It might be that you've got lasers on that cytometer that mean that you can excite fluorochromes more optimally. So if you've got a machine with a 488 and a 561 laser, you might want to be using the 561 to be exciting your PE fluorochromes instead of your 488 fluorochromes. You also need to know things such as if any of your lasers are collinear, because if they are collinear, you actually find you can't use some fluorochromes together. For example, FC500 from Beckman Coulter share detectors because their lasers are collinear, so you can't use PE-Sci5 and APC together because they use the same filters, and therefore it's important to know what your cytometer can do before you start planning your experiment. So just to summarize, what we've talked about is how we can get a single cell suspension through the fluidics of the machine. So we want to get them out so that each and every cell is being analyzed, so we can get the most from our data. We then put them through a focusing lens of the machine. We then steer that any fluorescence that is emitted from the cells through optical filters. We then can detect those photons with a PMT. We then look at that signal through the electronics of the machine, and then we can see dots on the computer. And that's the end of the webinar.
Thanks very much, Rachel. That was an excellent presentation. So we have a few questions from the audience, and just to remind you, if anyone else has a question, please feel free to post it in the questions box that appears on the right of your screen. Rachel, just to start it off, could you just briefly speak of some of the differences maybe between the main commercially available machines?
There are differences in the fact that you've got some that are clinical, so they've been CE marked, which means that they have been checked that they can be used for clinical samples. There's also different types of lasers. There are some that have, such as the Curie, have a set PMT voltage, and therefore you can just run your cells without having to set a voltage, and you can then just zoom into your data. So it has a 7 log decade dynamic range. It's important to kind of work out what experiment you want to do to then look to see what machines you have and what's actually available.
Could the minerals in tap water cause a blockage?
I've not seen minerals cause blockage, but when I have run cytometers on water, I have seen a little bit of mould, which our engineer wasn't too happy with, I have to say. I don't do that in these machines I've got now. I use PBS, but I have in the past used water.
I have a question here too. What is the minimum threshold value?
My machines are the BD machines, the LSR2 and the Fortessa. It tends to be set on 5,000, but I don't actually know what that 5,000 corresponds to as the threshold lowest level. On a caliber, it's the lowest level. The set level is 52.
Okay, so I guess it's just specific to individual instruments. Yeah. Someone has just asked, could you explain again the two lasers sharing the same path? So I guess just in relation to the collinear.
Yeah, some machines that have two lasers going through the same pathway. And what can happen is that if we have got fluorochromes, they could be excited by both lasers. So what we want to make sure is that we know what fluorochromes are going to be used. So if you've got an Acuria machine, it's actually got two lasers, it's got blue and a red laser, and they're collinear, but they're pulsing. So the red laser will pulse on and then the blue laser will pulse on. But machines such as the FC500, they're sharing a common path, therefore you can't use fluorochromes such as PSI5 and APC together because they both will be collected in the same detectors.
It says here, does it mean that side scatter is only in the direction towards where the detectors are, or it's in all directions at 90 degrees?
Light scatters all around, but most machines collect it at 90 degrees. Some machines actually collect it at different angles, but majority of machines collect it at 90 degrees.
How many events are considered optimum?
It depends on what machine you're using. Some of the electronics don't like to have too many going through. On machines such as the Fortessa, I would say probably I wouldn't go more than 10,000 events per second, especially if you've got multiple parameters, because the problem with that is the electronics of the machine are trying to cope with per second on, say, up to 20 parameters, and that's quite a lot for the machine to be able to do in one go. I know some machines theoretically can go a lot higher. I've never pushed machines too high.
I'll just read out the next one now. I was running a monocyte population. During the running of one sample, the screen was full of tiny small dots. Do you have any idea what may have went wrong? So I guess maybe what the small dots might be.
It could be that you've actually introduced air. So if you run your sample dry, it could be that you've actually introducing air, and that's going to then, you're going to suddenly see your screen filled with the dots. It is possible to actually save your data. You can actually use time as a parameter, and I think we've got a webinar I think it's Wednesday on time, which I'm sure we'll talk about how you can actually gate on time if you've accidentally got air into your system.
Do you think, is there a way to differentiate poor labeling versus low number of cells?
I think that's very difficult to know. I think it's always important to titrate your antibodies so that you know you're using the right amount of antibody for the right number of cells. And I guess try more than once to make sure that it's not just your cells haven't labelled well, or it could be sometimes an antibody that can be problems with antibodies.
Can the filters be changed to best select for the fluorochromes, or do fluorochromes need to be chosen to best fit the machine? So I guess, is there a way of changing in and out the filter sets?
Yes, most cytometers it's possible to take the filters out. Manufacturers tend to put in standard filters to the most common fluorochromes, but it is possible to change them. Sometimes you might actually want to change your fluorochromes used in your experiment so that you actually make your experiment easier. So you've got a multi-laser machine, you can use fluorochromes that are far apart, so therefore you're not going to get any problems with spillover.
Okay, actually that probably brings me nicely to the next question. Have you any advice on how to reduce compensation?
If you've got a multi-laser machine, then I would try and get fluorochromes far apart from each other. So say you've got 4 laser machine and you want to do 4 colours, try and get one of each of those. Try not to get fluorochromes that will overlap next to each other when you're doing an experiment.
It may be on the scope of this, but would you like to comment briefly on, to kind of discuss compensation, and do you think you have time for that?
Would you like to know about compensation?
The question is just returning compensation. I guess, can you briefly maybe, spectral spillover, really?
I'll just go back a few slides and I will show you some things.
Great, thanks.
So when we excite a fluorochrome and it emits, what we usually get is a plot, and it's more likely that this fluorochrome, for example, the one on the screen, will fluoresce yellow. But sometimes during the fluorescence process, it actually can come out with a different wavelength fluorescence. So we end up getting some photons that are emitted in the red. So if we were to collect that emission, we would collect it with probably something like a 580-40 filter. It kind of looks like a bit like a PE. But if we were then to look at, say, Psi5, we're going to collect that around about 670. We're actually, if we looked at the Psi5 channel, we're actually going to see a little bit of that PE emission in the Psi5 channel. And so what we need to be able to do is say to the machine, that's not actually from the Psi5 channel, that's from the PE. So we need to ask the machine to look at the PE Psi5 channel minus percentage of the PE. A lot of cytometers will do this for you. You run automatic compensation, and I'd always recommend using automatic compensation, whether it be on the machine or running it afterwards on post-acquisition software. Flojo can do compensation for you because it can do it much more accurately than we can. So make sure you run your single color controls, and if you can, try and avoid compensation by trying to get fluorochromes that are far apart.
Are there a minimum number of cells that you need to perform flow cytometry?
It depends on what you're going to be looking at. I always try and save as a minimum 10,000 events going through the machine, but it does depend. If you're looking at a rare population, then you need to make sure you've got enough of your rare population to make your results statistically accurate.
There's just a question here about absolute count. Can you tell us maybe why we're not able to do absolute counts on a standard, differential pressure cytometer? So
the flow rates of that are set on the differential pressure machines aren't exact. They've not really been measured, and though we go from low, medium to high, and they can be read in the manual, you can say what they actually correspond to. They've not actually been measured, so therefore we don't know if we run a sample, for example, 2 minutes on high, that we're actually running 120 microliters of your cells, because it also needs to take into account aborts going through the machine and things like that. So you're actually best to put in some beads. So you can get some beads where you know exactly how many cells per mil you have. So you add those to your sample, to a known amount of your sample. If you gate on those and save them to a certain number, then you can do some maths to work out how many cells per mil you had.
Sometimes two cells might pass the laser at the same time point. Will they be considered as a G2M population in cell cycle analysis? And if so, how would we control for that?
They might not show as G2M in cell cycle. What you would look at is the area versus the width, or the height versus the width. So when I showed the pulse, you can actually look to see whether or not we've got two cells stuck together, because we can compare the width to the height and the width to the area. So it's important, especially when doing cell cycle, that we do doublet discrimination of our cells.
Okay, so we just make sure we're just looking at single cells on our analysis window.
Yes.
Okay, so I think that brings us to the end of the questions and the end of the seminar. Thanks again, Rachael, for a very illuminating presentation and a great discussion. I'd like to also thank our sponsors, Novus. If you have any specific questions about Novus or the products, please contact Tom at novusbio.com. And finally, thanks to you, the audience, for taking the time to attend and listen in. If you enjoyed the seminar and would like to view the video recording of the session, please visit the seminars page on bitesizebio.com. It should be available within the next 24 hours. There you can also see the other webinars we've lined up for you in Bite Size Bio's Webinar Festival. So until next time, good luck in your research, and goodbye from all of us at Novus and Bite Size Bio.